All right, thank you, Ed, for that introduction. And actually, he kind of took some of my slides thunder.
Sorry.
But I was actually happy to hear that a lot of you have not heard of the Mission-Aransas NERR, because I'm going to give a quick background. Sometimes a lot of people have heard of us and they have to sit here and listen to me talk about what I love to do. And this is not working either, so we're just gonna do an old-fashioned. Actually, I know what it is. It's the. There she is. Got it? Yeah, I was like, I know what I can solve this one. So here is a map of the NERR system. So as Ed mentioned, there are 30 reserves part of the system. We are basically funded and kind of NOAA is kind of our leader in a lot of our doings and procedures and what we're doing on a daily basis. The mission of the reserve system is to practice and promote stewardship of coasts and estuaries through innovative research, education, and training using a place-based system of protected areas. So here's a map of our reserve here at the Mission-Aransas. As Ed mentioned, we are the third largest, so we're just over 186,000 acres. Within our reserve, we do have the Aransas National Wildlife Refuge. We have Goose Island State Park. Ed mentioned, going back. Fennessy Ranch, which is this kind of larger parcel of land up there. It is a privately owned ranch that we own a conservation easement on. So it's really an interesting and unique opportunity for us to be involved in land management of that property and see how a number of different land management practices, cattle, hunting, can all be done with an environmental mindset. A lot of our property and land is also open bay, as you can see, but then we also have the mangroves, Harbor Island, a lot of marsh habitat as well. Within each reserve, there are 4 sectors. So I am the stewardship coordinator. Ed, he may not have mentioned, is actually the research coordinator, as well as a number of other things here. And then we have education, which Lily is part of the education sector or program, and then coastal training, or we have a CTP coordinator as well. And each of us have kind of very broad kind of priorities or tasks, but we all do work, and it is recommended, it is encouraged that we work together. Hence why I'm here presenting, doing outreach. I work with Ed and his lab a lot, doing research in the swamp program, and then also the coastal training program is that coordinator really works to bring the science to the resource managers or to the decision makers, communities, mayors, city councils, stuff like that. They're the CTP coordinators who bridges that gap. But again, we're all kind of intermixed and mingle. So here, no offense, Ed, I'm going to focus on stewardship because that's my bread and butter. But here are just some of the things that I focus on with my program. Habitat monitoring is really what I'm going to focus this talk on. I also do a lot of marine debris research and monitoring. We at Fennessy Ranch, we actually are the ranch managers. So about 3 years ago, we approached the ranch, asked if we could, well, the ranch manager had retired. And so we, I thought it was an awesome opportunity. I still love it, but we took the primary role of managing that 3,300 acre ranch. And then we also work and do some restoration, outreach, and then I also am focused on invasive species control and monitoring. So today I'm going to talk about the mangroves. Here's just a photo of, this was obviously pre-2021. But one thing I did not put a slide in that I keep meaning to, and I forget. I just wanted to say that any of the research or any of the data I present on, I cannot do without the assistance of volunteers, my lab techs, We have graduate students, not only from UTMSI, but from Texas A&M Corpus Christi that come out into the field with us. And so all of this data and all of this research and monitoring could not be done without assistance. So I want to thank them and give a shout out. So what are mangroves? Mangroves are ecologically, mangroves are defined as an assemblage of tropical and semi-tropical trees and shrubs. Here in Texas, we really mostly have the scrub shrub. I'll kind of explain that here in a moment, but we have the black mangrove, and it's more of a short shrub black mangrove variety. But mangrove is a community composed of a plant whose species have special adaptations, and just two of those that are the biggest kind of known adaptations for mangroves is one, the pneumatophores, so those kind of fingers for the black mangroves. That's those finger things that stick up out of the ground. If you've ever seen a black mangrove, they allow, and here's a red mangrove, and it's the pneumatophores are the kind of like the rainbow shaped fingers going into the ground, but it allows these mangrove plants to live and survive in low oxygen environments. And then another adaptation is on the very first slide, actually, you saw there were salt crystals on the mangrove tree leaves. Mangroves are actually able to expel salt, so they can live in high saline environments and actually put salt on their leaves, which is a cool adaptation for these plants. But again, mangroves are not like a specific species or a tree, but they're actually thought of more as a habitat. And so they're defined by their ecology rather than their taxonomy. So mangroves and ecosystem services. So why do we care about mangroves? First, what is an ecosystem service? Quickly, it basically is the benefit that a habitat, so mangrove, salt marsh, oysters, how that benefits us as human beings. So it's a very human-centered way of looking at these habitats. But at the same time, when you're talking about resource management, trying to get money for restoration, you need to connect people to the habitat, and that's what we do with ecosystem services. So mangroves, how do they benefit us humans? One, erosion control. So their root systems really suck and like stick down and keep sediment kind of solid to the ground. And then they also can protect for storm surge following tropical storms or hurricanes. They do storm surge protection. They provide a number of, they provide habitat for fish, bird species, ecotourism, carbon storage as well. So mangroves can do a lot of different services for us humans. It's basically what it comes down to. And then climate impact. So climate change, there are impacts. Climate change can have impacts on mangroves, is what I'm trying to say. But the climate stressors, the number one stressor for mangroves is going to be sea level rise. So as the water is rising, it will impact these mangroves. They do live in low elevation and can be inundated, but they do have periods where they need to be dried out. There are certain water levels, they can't be underwater for days and weeks at a time. So as the sea level rises, they will be impacted. Hurricanes and storms can hurt, obviously hurt a mangrove. They can rip them out of the ground. They can defoliate them and cause mortality. And then changes in precipitation, whether it's drought increases salinity in these habitats, which yes, a mangrove can live in high saline environments, but there's a tipping point. And then also too much fresh water can bring in nutrients, pollution, sediment, heavy debris from like a river flood or something, which can be detrimental to the habitat as well. And then obviously anthropogenic stressors, such as pollution, deforestation, or basically removing of the habitat and sedimentation, you can bury the mangroves. And then put both of these on top of each other and you have compounding effects. So just a quick slide on where mangroves are. in the world. As you can see, they basically can be found across the globe. Here in the Gulf of Mexico, and this is actually looking at species numbers. So there are close to 50 species, different kinds of mangroves in the world. Species diversity wise, you can see the hotspot is kind of over here off the coast. I don't want to use the pointer because I don't know what's going to happen. Off the coast of like northern Australia. But here in the Gulf of Mexico, we basically have 2 to 3 species. And here in Texas, we basically have 2 main species. The black mangrove is by far the dominant species. The red mangrove was becoming more and more prevalent prior to 2021's freeze. And you can see that there are hotspots. And what they mean by hotspots are the black mangroves or the red mangroves are actually expanding in habitat and ranges. And so the black circles represent kind of those expansion hotspot areas. And you can see that Texas, basically the entire coast, is a hotspot for mangrove expansion. Okay, so one thing I was going to, not only am I going to talk about what we're doing here at the Mission-Aransas when it comes to mangrove research, I'm going to kind of talk about what 5 other reserves that are part of the system are also doing for mangrove research. So a lot of it overlaps with us, and then a lot of it provides a different view of mangroves and habitats than what we see here in Texas. So I kind of thought it was interesting. We actually just presented all of this research in slides at a presentation or a conference in New Orleans before Christmas, and I asked the researchers if I could kind of steal their slides and give you guys kind of a view of what other reserves around the system are also doing. So here in Texas, like I mentioned, this is all black mangroves. We are very dominant. We are monoculture black mangroves, scrub shrub black mangrove, typically 3 to 4 feet tall. We did have a few individuals that could be, 8, 10, 12 feet tall, but they were very kind of isolated in specific areas. For the most part, I could stand up in my habitats and monitoring areas and see where other people were for, miles around. I did mention there were a couple red mangroves. They were, once you became familiar with what they looked like, you could kind of start pointing them out. They're a little different green. They can be taller. There were maybe 2 or 3 here on the coast that were taller. Red mangroves can get up to 30 feet. They're forest species. So there are a couple that, were 10, 12, 15 feet tall, but for the most part, the red mangroves we were starting to monitor were actually still pretty 1 foot to 2 foot tall. And they were mostly along the edges, along like ship channels or creeks where you could see how the seeds came in, deposited, and so you had a lot more red mangroves along those channels where the seeds were able to deposit and get into. Here's just another slide. So this publication came out in 2022, so pretty recently, and basically what we did was they surveyed every researcher along the Gulf of Mexico that was doing mangrove research, and they had us look at all of these little boxes and said, is there a mangrove in that box? If it is, turn it green. If not, turn it orange or brown. And so this is the most recent, as of 2021 pre-freeze data that you have for where mangroves exist in the Gulf of Mexico. And again, you can see we have this hot spot in this area with expansion also along Louisiana and North Florida. And we are, one of the things at the Mission-Aransas that we really are looking at with that hotspot is the transition. So mangroves are expanding, that means we're losing something else, right? If we're gaining mangrove, we're losing something. That something is mostly salt marsh habitat. And to us, that can be we're not sure what it means. It can be very concerning for people that are trying to manage salt marshes for whooping cranes. It can be concerning for fishermen that need a salt marsh habitat for fish nurseries, but then also mangroves have different positives. So that's one thing that we're trying to figure out is pros and cons to both habitats, and then also how the transition is going to look and how it's happening in the reserve. Here is a publication from Paul Montagna's lab in 2011. And basically with this slide, I just wanted to show that mangroves here, this is Redfish Bay. For any of you guys, it's basically that bay in between Port Aransas and Aransas Pass, the Harbor Island Causeway. You have Redfish Bay North and Redfish Bay South on your two sides. But this basically shows that we've had mangroves. They know at least back to the 1930s, but what it shows is that it's cyclic. You have mangroves, they expand, we have a major freeze, they contract, they grow, we have another freeze, they contract. So we know that there's a pattern to the mangrove growth and establishment here in Texas. So they're not invasive, they're not unknown to be here. They're a native species, it's just not sure, you know, how much area they should or have historically claimed. One thing, so going into monitoring these mangroves. So part of the Mission-Aransas NERR, and actually the NERR system, our backbone, what we call our bread and butter, is what we call the system-wide monitoring program, or SWAMP for short. So basically what we do, this program has 3 components. We have an abiotic, a biotic, and mapping. So abiotic is basically looking at your water quality and weather stations. Biotic is the mangrove marsh monitoring. We have phytoplankton monitoring, zooplankton monitoring. And then the third component of this SWAMP program is mapping. So we do a lot of habitat mapping, change analysis of the maps, stuff like that. Or we should be doing that. It's coming. It's on my list. But what I wanted to show is within the reserve, we have these 5 dots. So the purple and the black represent 5 of our water quality platforms. And then we also have surface elevation tables and then vegetation sites that include mangrove and marsh monitoring. So here's kind of where we are within the reserve system. And here's just an example. So the water quality data, everything is live and online. So you can go on to the website and you can actually get 15-minute interval for all the data we've collected. Water quality, not 15-minute vegetation data, but water quality data from basically 2008. We do have some gaps. Obviously, Harvey took out a couple of our stations, so we have a couple of gaps there. But this is just kind of one of our big products and items that the reserve system is really proud about. And what makes it system-wide is that we have these standardized protocols. So the way that I go out and monitor the marsh is the same way that they're going out in New Hampshire and Alaska and Washington State to do their marsh monitoring. So what I'm measuring can be compared to all these other marsh sites across the country. So it's standardized, it's really strong. It makes the protocol and the monitoring strong. And that's the same for the water quality stations as well. So all of them go out and maintain the stations the same way. They take the 15-minute intervals, data quality is all the same. You QA/QC it, and we're all following these same protocols. So what is the vegetation monitoring protocols? What's the point of them? What are they designed to do? So the first thing is they're there to quantify vegetation patterns and then their change in space and time. So because we're out there monitoring, we're able to actually see the impacts of the freeze, right? We've been monitoring mangroves since 2011. So we have 10 years of data before this freeze hits. And then we're funded to go out there for, as long as the reserve is here, to go out and monitor and keep monitoring these mangroves. Same thing, it's not just the freeze, it was with Harvey. We are in the middle of monitoring our elevations and our vegetation when Harvey hit. So literally some of the marshes, I was out there on Monday, the storm hit Friday. I was able to get back out there a month later to see exactly what impacts that storm had. And it's because of these protocols and monitoring methodologies that we're able to have that data and that monitoring. With the protocols, we're also consistent, like I said, with other monitoring protocols nationally and worldwide. And then we're consistent over a wide range of estuarine sites and habitats for reserve-specific purposes. And then also the fourth is that the data can be used not just for monitoring and understanding changes, but Lily and the education program can bring it out to classrooms and use it as education program or education resources. NERS and NOAA, we can bring it to our legislators and our funders and say, hey, look what we're doing. You're funding this great research, this great work. We're protecting all of this habitat, and this is why. So again, here is our reserve, and here are the two sites that I do specifically monitor mangrove monitoring at. So here are what the sites kind of look like. So here, and I'm going to focus today mostly on this Harbor Island. So this historically has been mangrove. We have been monitoring Harbor Island or Lighthouse Lakes. I might use those interchangeably. It is Harbor Island, but there's a lighthouse there, so I usually say lighthouse. We've been monitoring that site though since 2011. And then Mud Island West was actually established right before I came on board, and they established that site, and that site has actually been using the marsh monitoring protocols. So previously it had been a marsh. They put the vegetation there specifically to understand the transition from marsh to mangrove. And so we recently, actually right before the freeze hit, we established mangrove monitoring as well. So they are two different protocols. And so at Mud Island West, we are doing both protocols, methods, while at Harbor Island, it has always been mangrove, and we only monitor the mangroves. Here is what Harbor Island basically looks like. So we have 5 transects numbered 1 through 5. I just want to go back. And can I? Oh no. Uh-oh. Is there a? Yes, I found it. Yeah. Okay. So here is transect 1, 2, 3, 4, 5. And so this is what I'm going to call the Lighthouse Lakes channel. And then right here is Lydian channel. And so in this schematic, we have the Lighthouse Lakes. So the lighthouse might be here. Lydian channel is going to be up here. And we have those 5 transects. And then in those 5 transects, we have these 4 10 by 10 meter plots that have been established. And then within each 10 by 10, we have 3 1 by 1 meter plots also. So that means for each transect, we have 12 1 by 1 meter plots and 4 10 by 10s. And the 1 by 1s are randomly, they're established, but when they were established, we literally went out there, chucked a 1 by 1 meter plot, and wherever it fell, that was where we put the plot. So here's just kind of an idea. The 10 by 10, here's the 4 corners. There's a pole, a human, another pole. So that's 10 by 10. And then also in the 10 by 10s, So the original protocol, I will say, was the mangrove protocol for the NERR system was written for those large red mangroves, so the trees. And so they wanted us to tag and identify every tree in that 10 by 10. That's not going to happen. We laughed. So what we did was we went out there and we tagged 5 trees. And so we were like, we will do those 5 trees and we will get very in-depth measurements on those 5 trees. And that's what you'll get from us. And then we have the 1 by 1s, like I said. So what measurements are we taking? So in these large 10 by 10s, what we normally do on a typical field day is we'll arrive at a 10 by 10. I'll send 4 people to the 4 corners and we'll sit there and we'll scream at each other and we'll say, hey, we have a really large, channel here. I've got no plants here, but a lot of pneumatophores, and we'll scream and we'll discuss percent cover. And we're not just looking at percent cover, and that's how much plant coverage we have in that 10 by 10. We're not just looking at mangroves. We do record any species we find. So any succulent, any Spartina grass, we'll record that as well. And then we go and we search for our 5 tagged trees, which is actually pretty easy. I don't know. I guess I'm just familiar with where they were because we don't have any of them anymore, sadly. But we did these 5 very intensive measurements. So we would do canopy height, how tall they are. We would look at how thick their trunk is. We did clearing height, which is basically the distance between the ground to the very first branch of that tree. And then canopy wide is what is the widest part of that tree. Canopy narrow is, and then canopy narrow is the narrowest part of the tree. And then the canopy offset helped us determine basically how round that tree is. Is the tree a circle? Is it an oval? How, kind of what's the shape of that tree? And so those were the measurements we did for those 5 individual trees. And then in our subplots, those 1 by 1s, we would do percent cover. And then we measured every tree and sapling. We did height, and then we also did how the trunk diameter, so how thick that plant is. And sometimes we had 150 trees in or individuals in just those little 1 by 1s. And so this is actually some of the data from that transition site I was talking about. So that Mud Island West that we established, and we actually did our marsh mangrove or our marsh monitoring protocols. And so this is just kind of, it shows the data on how that site had really transitioned. So at the beginning in 2011, you had some grasses, you had good succulent species, so that lighter green. And then this darker green is the mangrove. And as you can see, as the years go on, the mangrove really takes over the succulent and grass species. So then what happened in 2021 and what is happening now? So I'm sure many of you know, in February 13th, basically Valentine's Day, we had a major freeze. And basically this is what happened to the mangrove habitats. So this was a photo from 2020, and then that was a photo basically 1 month following the freeze, so March 2021. And so we really wanted to know, how are the mangroves going to come back from this? Are they going to lose their tree or leaves and resprout? Is it going to be new growth? And so we're in a great position, right? Because we have these monitoring protocols to get back out there and monitor and see what's happening. And so this is what we have so far for at least Harbor Island. I have not worked up or looked at the Mud Island West data, but for Harbor Island, that Lighthouse Lakes area, this is what we have. So you have about anywhere from like 50 55, 75% mangrove coverage in all of our plots. And then the freeze hit. And we went back out that spring. So normally we are not monitoring in spring. We usually go out in fall, peak biomass season. But we did want to go out immediately after the freeze. So we went back out, collected spring data, and then went again in the fall. And this right here is basically, there were plants with green material, but it's a lot of resources state that even if your tree has some kind of freeze burn or brown leaves and green leaves, they're going to lose their leaves. They're damaged. It's just taking longer for those plants to lose their leaf material. So that's what you're seeing here. Are these trees just hadn't lost their leaves. But of course they did eventually. And one thing I did want to point out is that Harvey really did not impact. I know some people ask me about Harvey. Harvey really did not impact the mangroves at all. When we went out there, just a quick note on Harvey, the taller trees had like defoliation, but the shorter ones didn't. And that was basically because the storm surge protected those trees. So if you think about it, they're short, they're only 3 to 4 feet. You had all this water coming into the system. They were underwater. So the trees and the damage we did see were the parts of the plants that were still being whipped around by the wind and the hurricane strength forces. And so we saw damage on that part, but anything underwater was fine. And most of our mangroves here were underwater. So Harvey did nothing for the mangrove. However, Uri did a number. So as you can see, we're seeing some recovery, by the way, and this is a big question, right? Is if we're losing all the mangroves, what's the salt marsh species going to do? Are we going to see salt marsh start increasing and replacing or growing in the areas where mangroves used to be, or is it going to come back as mangrove? And so we're starting to see some recovery of salt marsh, very little, minimal recovery of mangroves. But if you look at it kind of at a different way, so this is looking at those 5 transects. So I separated out the data into transect 1, 2, 3. So basically what I wanted to do was look and see if like, basically is the distance to the Lydian channel, that kind of deeper, you're closer to the Gulf of Mexico water, source of water, if that had any impact. And I'm not seeing anything so far. Very little recovery. Again, your salt marsh species, you can see in 2021, 22, but no difference in your transects. And then this, I looked at whether or not the plots that were closest to the Lydian channel. So again, those channels, you get your seed source is closer. So new seeds can come in. Water circulation's better the closer you are to the channels. So again, like plot 1, plot 2, 3, and 4, and the 4th is the farthest one back into the mangrove habitat. And what you're seeing is actually you have a pretty good recovery of your salt marsh species on that edge. So you're seeing kind of an edge habitat or an edge effect with salt marsh recovery. But again, you're still not seeing much, if any, mangrove recovery, at least in 2022. So now I'm just going to kind of show some overall photos of what is happening. So this is that Mud Island West site. And you do see some mangrove recovery in October 21. Again, it's along the edge. So we're seeing not just salt marsh, but where the mangroves are recovering, it's also kind of on the edge as well. 2022, you have some recovery. Here is recovery with salt marsh in May. This is Mud Island West as well. And then here is Harbor Island, June. So just over a year and a half post storm. And it's really what we're seeing is just this real spotty recovery. We're not seeing tons of recovery everywhere. It's really spotty and sporadic. And so, like I just said, it's localized and spotty. It's mostly on the edge. And one important thing to note is that any growth that we are seeing, we're noting that it's new plants. So it's not the old plants producing new leaves or rebudding from the base or the trunk. It's actually new plants coming in, new seedlings, stuff like that. So what are we going to do moving forward? We're going to re-implement the full monitoring protocols. So we're going to keep going out there every year in the fall. We're going to tag new trees. I did that this fall. Actually, a lot of our plots did have 1 or 2 trees that we were able to get tagged. So we'll start following those trees on an annual basis. And then we're starting to look and think about mapping where the regrowth is happening, what those maps look like, how much acres, stuff like that. But then there's also a lot of additional monitoring and research. So why are the plants recovering where they're recovering? Why are they not recovering everywhere? You know, you can look at down one channel and see a couple mangrove plants here and nothing on the north side. So it's very sporadic. Is there a reason for that? What's the random, like, you know, in nature, nothing, you like to think nothing's random. So we always want to get out there and trying to find the reasoning. Okay. And then in the Mission-Aransas, we also have a number of different researchers, partners looking at the freeze. So researchers from A&M Corpus, USGS, A&M Kingsville, TAMU Galveston, a ton of researchers kind of looking at the impacts of the freeze, the recovery, and the impacts of the expansion. Other research, we have carbon flux. So this is actually a really cool project. Dr. I'm just going to call her Dr. Ashley, from UT Austin has this huge tower in the middle of our mangroves. And she placed this tower, one here in Texas, one in Abu Dhabi. Yes, thank you, Lily. And then one in Panama. And what she was looking at is how carbon, so like respiration, photosynthesis, changes on how dry or arid the environment is. But she put this instrument in the mangroves prior to the freeze. And so she called me like the week after the freeze and was like we've you know, redlined like the mangroves before she could see these like daily, you know, respiration rates of this whole habitat. She could see like the respiration, the carbon going into the system during the day when the plants are photosynthesizing. She could see them like, you know, releasing oxygen at night. And then after a week, she just said like, we're seeing nothing left in the habitat. So it was really cool to see that data and to hear her perspective. And she basically from Austin was like, you know, we can see it here. And then we're also looking, or Steve Pennings and Dr. Anna Armitage are kind of like the head gurus on looking at the impacts of the expansion and the pros and cons to the 2 different habitats. So quickly, I'm going to talk about these 3 other reserves, the one in Rookery Bay, Apalachicola, and then Hawaii, and kind of what they're doing. And I should wrap up here pretty soon, actually. So in Rookery Bay, Jeff Carter, and these are, I will say Jeff is another stewardship coordinator. So he's this SC for Rookery Bay. And they are just south in Naples, Florida, where they have the red mangrove forests. So they have a completely different habitat of mangroves than we do. But what they're, one of the really cool projects that they're involved in is this collaborative science research monitoring that they're doing with Hobos Bay Reserve, which is another NERR, but in Puerto Rico. And they're looking to understand the links between degradation, recovery, and community benefits, and understanding the factors influencing recovery from hurricanes in Puerto Rico and Florida. So both of those areas have been hit multiple years in a row by multiple storms. And so they're really wanting to look at the recovery of these red mangrove habitats and the impacts of these storms and repeated storms on their habitats. And then he also has this massive 225-acre mangrove restoration project. So they have a road running through a historic mangrove forest. And what they're doing is they're putting in these culverts to increase water circulation. So they found that they cut off a mangrove forest with that road and cut off the circulation, the water circulation. And so this mangrove forest basically is degraded, died. And so they're trying to improve the water circulation and then go back in, replant and regrow this large 225-acre mangrove forest. And again, this is in Rookery Bay, Florida. In Apalachicola NERR, so Panhandle, Caitlin is also, or she was the SC, she just left. But she was doing a lot of the same stuff we are. So Apalachicola is again at that same latitude we are, similar latitude. So they're in a transition hotspot zone also. And so they do a ton of mapping of where their red mangroves are and where their black mangroves are. But instead of having like vast areas like we do, she really does just have individual plants popping up all over her reserve. So where we might be mapping polygons, she's going out there and just mapping like individual points of mangroves. Again, here's kind of her mapping efforts. And she's mostly focused also on getting baseline spatial data, characterization of the habitats, where the mangroves are, where they're not, why they might be where they are, why aren't they where they're not. Looking at growth and recruitment of mangrove into marsh habitats. And they're obviously using this long-term monitoring protocols. Her and I actually are on a number of different projects and publications together. So we do a lot of gap analysis and what future research is needed for mangroves, especially in these hotspot areas and the transition zones. So we have gap analysis we do a lot of. Again, the spatial and time connection of mangrove habitat growth and restriction, distribution and gap knowledge, detection and monitoring using UAV, so using drones to go out there and be able to actually fly and get images, and then have a computer tell us like, hey, that's a mangrove plant, hey, that's an oak. Just for example, being able to use computers and drones to tell us where the mangroves are. And then impacts from freeze events. And so one of the publications we have out right now is looking at how that winter storm Uri actually impacted the entire Gulf of Mexico. So you have researchers monitoring the mangroves here, Louisiana, Mississippi, Alabama, Florida. And so basically the freeze happens and you can like hit a button and you like activate the mangrove monitoring network, and everyone goes out, they take temperatures, they take their mangrove measurements. And so you all have this like one time stamp data, and we can do a Gulf-wide assessment and analysis. And in Hawaii, they do have mangroves. And Kawika is actually the director of the Hawaii Reserve. Hawaii is the second newest member of the system. It's the one I want to go to most, but someday. But the really cool thing I wanted to mention about the Hawaii Reserve is that their goal is to support biocultural restoration through indigenous community-led stewardship. So their reserve is heavily involved and heavily dependent and kind of included in elder guidance. So they have a lot of history and historical knowledge, and they bring in a lot of the indigenous practices, habitat restoration, monitoring, stuff like that into the reserve, which I think is really cool. So I just wanted to, and they always make a point whenever you talk to them to honor their elders. So I wanted to make sure I did that for him. But in Hawaii, mangroves are actually invasive. So they ruin their water quality. They are not good habitat for native species. They're excellent habitat for invasive species. They cause issues with water quality. The roots damage the natural infrastructure. The biodiversity is much lower in mangrove habitats than in their natural habitats. And then they're not providing fishing, timber, or livestock. So those ecosystem services I talked about that mangroves provide for us, they are not providing what they should in Hawaii. And so Hawaii, and here is the reserve. Well, the reserve, I'm not sure what their boundary is, but this is within the reserve. But you can see right here, this is a large red mangrove forest. And so this is 10 years ago. And so they're starting to work at removing it. But again, here you can see those prop roots and pneumatophores into the system. And here you can just see these are red mangroves. They're huge trees. They're like 90 feet tall in Hawaii. And they're starting to take them down and remove them and bring back the habitat. And so you can see what they did here. So this was that photo from 10 years ago. And you can see they've cleared all of this area of the red mangroves so far. It's an ongoing project in Hawaii. But they've already seen improvements in the aquaculture, their water quality, sediment retention that can help grow food. And then they also have a number of native and endangered bird species starting to reuse, these beach areas, these shorelines have some native birds coming back and endangered species. So to quickly wrap up, I just want to talk about in general, the threats to mangroves are development, shipping channels, chemical spills, and this is kind of Gulf-wide. Hawaii, it's completely different. But for the Gulf and where mangroves are native, you have development, shipping, chemical spills are threats. The weather events such as freezes, hurricanes, sea level rise, visitor use. So over, a ton of people walking through the mangroves can cause some issues. A ton of researchers walking through the mangroves causes issues. Coastal wetland erosion, and then just a lack of awareness. So a lot of people are not sure whether or not they should cut down mangroves, leave them. Some people will pull them. They're understudied. The people that are really wanting those salt marsh habitats for the whooping cranes are all about pulling them, freezing or burning them, doing whatever is needed to stop the mangroves. But, there's pros and cons. Challenges to monitoring mangroves or managing mangroves are the monitoring methodologies. So I discussed how black mangrove density is way different than red mangrove density. The red mangroves are very spotty, at least here in the Mission-Aransas and Apalachicola. And then understanding and monitoring that expansion into the marsh habitat. Defining the impacts and implications associated with sea level rise, climate change, and the mangrove habitats is a challenge. Limited historical baseline data, and then unknowns. Sorry, I need my glasses. Unknowns at a landscape level. So what is happening on a landscape, not just like individual, we had a freeze here, but on a more landscape, Gulf of Mexico, regional, or Texas-wide, what's happening with the mangrove habitat. And so any questions? And thank you guys for listening. and being a good audience.